invite Dr. Rosalind Moran. Thank you. We'll continue to... here we have Rosalind, who is a imaging specialist. She is from Dublin, at the College Dublin in Electrical and Electronic Engineering, and at the Wellcome Trust Centre for Neuroimaging at the University College of London in neurology. She is currently working on neuroimaging and computational neuroscience, eminently suited to lecture in an auditorium like this, wherein engineering scientists intensely interact with the neuroscientists. There's been a long record of the electrical and electronic engineering department working on imaging and signal processing with, for example, NIMHANS, and now of course with the department of the CBR. So, it's very appropriate that she is here with us and we are presenting on imaging and that predictive coding. Did I read that right? Predictive coding, aging, neurobiology, and vulnerabilities for neurodegenerative disorder. Firstly.
Thank you very much.
Thank you. Good luck.
Thank you. Good morning, everybody. Thank you for the invitation here, and it's particularly because of the interesting mix here of computational neuroscience from engineering meeting clinical neuroscience that it's wonderful to be here. Thank you, Dana, for your message of solidarity with people of Paris and for your excellent talk. And I'm going to build on some of those ideas we heard, like the posterior anterior shifts in aging and the Harold model, but take you through a more computational perspective on aging, specifically built around ideas of predictive coding. And I know there's a poster on predictive coding in schizophrenia, so what I'd like to take you through is a theory of predictive codes and how that might suggest some potential fault lines in circuitry as we age. So I'll try and do it without too many equations as well, because it's a little early for those. But just to give you a general idea of what I do, as you heard, my background is in engineering and computational neuroscience. And our lab uses something called dynamic causal modeling, developed with colleagues at the Wellcome Trust Centre for Neuroimaging at UCL, to look at effective connectivity in the brain. So how do circuits interact and how are they modulated by particular experimental modulations and paradigms. And I'm interested in aging and also dynamics of brain evolution at timescales, so as we sleep, and even shorter timescales, so how do dynamic interactions lead to complex and simple behaviors. So this is a general framework in which I study aging and neurodegenerative disease, and today I'm going to take you a little introduction to that framework. So the approach is slightly different to, say, functional connectivity analysis. You try and build a detailed model of brain dynamic circuitry and try and build into that model from a basic perspective priors on what other neurophysiologists have learned over the last several decades on what really constitutes dynamic information flow in the brain through ion channels, receptors, and neurotransmission. And we try and build that into a framework so we can study human neuroimages. Then I'm going to review again some of the models that you heard in Anna's talk, so models of compensation, differentiation, and scaffolding, and then introduce you to a theoretical idea based on predictive codes of why these activations could move to the front and move across hemispheres. And then I'm going to present some work from my lab that has some support for these predictive codes in older brains, specifically in the context of learning and memory paradigms using fMRI, EEG, and dynamic causal models. Okay, so to build a generative model of brain dynamics, the general idea is to try and uncover synaptic or unobservable events in animals using neuroimaging and electrophysiology, and to that build a mathematical microscope for connectivity or network assistance. So the state of play is to build a biophysical model. So these you'll be familiar with perhaps at a cellular level, We can also build them at a more macroscopic level, and to look at columns of activation, columns of interacting cell populations that might lead to an activation. This here is from a magnetoencephalographic recording of theta oscillations. And then we're going to measure these activations. We can't measure synapses or anything at the mesocolumnar scale from neuroimaging, but we can measure the activations, and we'll use Bayesian statistics to try and infer the synaptic properties associated with the behaviors and the activations. So we're sort of trying to close the loop with Bayesian inference so that we can uncover things that we can't really see, but we can model and test different hypotheses of how ensembles are connected and how they interact in a given task. So generally the models are built with this predictive coding framework in mind. What I have here is just a picture of a synapse, Neurophysiology 101. So we're going to model excitatory synapses with glutamate, exciting AMPA and NMDA receptors. We'll also include in our models GABA, so we can look at inhibition. And we want to build them into a framework that's suitable for predictive coding. And this picture here on the right is a famous image from the first issue of Cerebral Cortex. It's an image of tracks in monkey visual hierarchies. So it starts around the LGN and thalamus, and then goes into V1, and then more progressively complex regions of extrastriate cortex into all the way up here we have the hippocampus. This was a demonstration that sensory systems are hierarchical, so they essentially build up from simple to more complex objects and recognitions and actions. And so using this in mind, we can try and use DCM, or we can see that if signals travel up as well as down the hierarchy. So it might be that complex information flows down and simple information flows up. And so how might that affect aging and brain dynamics? So as I mentioned, in our models, we'll have glutamatergic excitatory neurotransmission and GABAergic inhibitory neurotransmission. And we'll also allow for changes in synaptic gain, which we think is under the control of neuromodulators in our models. So things like dopamine, like we've heard about, noradrenaline, acetylcholine, and serotonin, all important for aging. This is a picture to outline the general dynamic causal modeling framework. And here is the sort of mesocolumnar level that I just described, where we're looking at sources interacting in the brain, and we do so using just a set of ordinary differential equations. So in the EEG and MEG context, we can have quite a detailed neuronal model, which I'm going to show you in a little bit, where we're operating at synaptic timescales. Obviously, we can only apply it to limited regions of cortex that are amenable through EEG and MEG, but in fMRI, we have a more simple neuronal model that describes the dynamics of interacting brain regions. And then we pass these dynamics to the observation level. So we know, for example, in fMRI that we don't measure neural activity directly, but see it through the hemodynamic response. So we use a balloon model here. It's a little simpler in animal electrophysiology, in MEG or EEG. We can use these neuronal models and pass it through a linear gain matrix, so we can see if our three little areas are interacting here in the brain, what does it look like on the scalp or in the sensors at that level. So modeling these brain waves, or indeed activations generally, is, as I said, this sort of closed loop process where we propose some model with a structure of parameters, and we want to make that model useful in terms of understanding aging. And then, given our empirical data, we use Bayesian regression to try and optimize the model structure or model parameters. And it can generate things in EEG and MEG, like oscillations like in different bands, theta, gamma. And it can also generate event-related potentials, which I'll go through in a little bit when we talk about our empirical data. So just to get into a little bit more on how much neurophysiology we build into these models of human brain imaging, we use something from statistical physics called mean field theory, where we use the properties of tens of thousands of neurons and approximate them with our average response. So these are sometimes called neural mass models in the literature. And it's a neuronal source model where, as I mentioned, we want to build hierarchies. So hierarchies in the brain, as you saw in Feldman and Van Essen, send forward signals up to progressively higher regions, and these higher regions send signals back to simpler regions. And importantly, they occur in different layers. So in our model, we're going to have this laminar structure which includes the sources and receivers of bottom-up or top-down connections, which is important when we're going to talk about aging. So in the model we have this layer 4, which is a nice stellate cell layer region, and this receives generally thalamic input and input from lower level regions, so it travels up the hierarchy. We also have pyramidal cells in the infragranular and supragranular layers and inhibitory interneurons all around them. And we really do describe these populations with parameters that connote intrinsic connections, where we look at the weight parameters and time constants of the different interactions. And also, as I mentioned, we embed it with hierarchical structures. So we can look at extrinsic connections that come bottom up the hierarchy or top down the hierarchy. And it's this sort of model that we then use to measure or to model an average dipole, which we'll call an activation when we look at our MEG or EEG signal in a particular part of the brain. Okay, so I won't go through the state equations in detail, just put them up iconically to say that basically there are these parameters that I can change, so the conductances at different synapses in this model, so we can change these parameters in the model to give us different data features. And so the idea of our DCM, or dynamic causal model, is to optimize the set of parameters for each individual person so that we can measure things happening within a column, for example, the relative strength of inhibitory interneuronal synaptic weights to excitatory synaptic weights within a region, and also look at areas from region to region, so the hierarchical bottom-up or top-down connections. So our parameters, as I said, really do model connectivity. So this is a detailed model of brain connectivity, which is a little different to the functional connectivity measures where you can be a little more agnostic to the mechanism. Here you really have to write the mechanism down as a set of equations. This is reasons why some people like or dislike DCM. I find it useful just for looking at the hierarchical interactions between brain networks. So there's a selection of intrinsic architectures, and just if you're interested, these are implemented in SPM, which is statistical parametric mapping, which you might have used for fMRI analysis. So this contains a suite of modular architectures for neuromodeling, where if you have your own favorite Hodgkin-Huxley embodiment of some brain tissue, you can switch in and out. It's written in MATLAB, so it's quite modular and user-friendly, so you can build your own models and use the inversion techniques or whatever else we've provided. So overall, what we end up doing is we have this interlaminar connections and set of neurons which have dynamics that evolve over time. Then we go to an experiment, and we know that, say, 6 active regions are active in our particular task. This is an example of regions that might be active in a mismatched negativity, which I'll show you in a second. So we might have regions of Heschel's gyrus in some auditory task responding, as well as regions of the temporal and frontal lobes. And what we can do is connect these little modules or models together so that when we ping it with an input, it'll generate these brain responses that are commensurate with real empirical data. Okay, so that's the framework. Does that make sense so far? Like some heads not? Good. Okay. Finally, just to get to the model inversion, and this piece one could spend a whole talk and career on, but it's if you're moving from a model to the parameters and inference on the model. So how do I know if my model is correct? And really you have to have a couple of hypotheses and compare them. So we define our likelihood model there, what I just showed you. We have some priors which we think can be neurophysiologically plausible, like the receptor time constants or the level of excitation to inhibition within a particular region. And then we do something called a free energy maximization, where we try and maximize evidence for a particular model by changing the parameters of that model. And then what we always do is something called a Bayesian model comparison, where we can connect regions differently, or we can put in different cell types, and we can say, is there evidence in my data for or against this model? And if so, is that evidence strong? Or do I need to come up with a new hypothesis, because basically my relative evidences are all flat? So it's a good test of whether you have good hypotheses as well. And then once we've selected our best model, we can then do inference on parameter space. So is an aged brain more connected bottom up, or is it more connected top down? You can probably guess it might be the latter. So we validate these models, and we've spent some time on that in the last number of years. This is not related to aging, but it's a nice demonstration of the technique and the technology. This was a study looking at channelopathies, which you might have seen made famous by this popular science book on NMDA and encephalitis. So there are some people walking around with remarkably few deficits who may not have a particular GABA channel or may not have a functional NMDA channel. And so they offer really good tests of our DCM. So if there's an ion channel absent in someone's brain, can we detect it with the models that I just described? So our aim in this particular validation study was to see could we identify channel abnormality in 2 rare patients with mutations in genes encoding for potassium and calcium channels. And we compared them to 100 lifespan control cohorts and compared them to the genetic channelopathies and used MEG to measure responses to auditory tones, the auditory Opwell task played into the ear, and we're modeling ion flow with our dynamic causal model. And so we have neurotransmitters and ion channels, so glutamate and GABA, as I mentioned, and we have different ion channels, sodium, potassium, calcium, and chloride. And remember, our 2 patients have particular genetic mutations which give you a loss of function in potassium and in calcium. And we played this auditory Opwell task where it goes beep, 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 boop. And what you see, and this is typical in EEG as well as MEG sensor data, is that you get a negativity to the Opwell. So this negativity here is in green, whereas the response to the standard tones is in blue and is much, much less. So we used this to validate our model, and we were happy that versus our 100 control cohorts, here I'm plotting parameters of our model that show the connectivity of different ion channels. You could see that the potassium patient was identified as abnormal, and calcium patient was also identified as abnormal compared to our healthy cohorts. So with this information, we're hopefully a little more assured that our models have something sensible to say about connectivity in the brain at the level of synaptic transmission. So how does this relate to aging? It seems a little perhaps tangential to this point, but I think they have real strong utility in studying the aging brain. So this is going back to Anna's talk. I've just highlighted a couple of models on cognitive aging that are predominant in the literature. So the hemispheric asymmetry reduction model in old age, or the Harold model, which is sort of synonymous with the de-differentiation hypothesis. And there are also other models called the STAC model, so the scaffolding theory of aging cognition. And I drew this myself, so I have to now put it into every talk I ever give. And it's basically to describe the type of activity that you'd associate with a young brain versus the type of activity you'd associate with the old brain. And in all of these models, basically what we have is a young brain that works something like this, and an old brain that works something like that. So predictive coding, what I'm going to show you next, I think with DCM allows you to explain why these patterns occur as they do. So young brain, nice and forward, nice and lateralized, old brain, delateralized, and predominantly frontal. But these models, the Harold model, the PASA model, I would argue are not real models. They're more descriptions of data features. For a model, you have to be able to generate data, so build falsifiable hypotheses. So I think we can get that a little closer to a real model if we look at some Bayesian predictive coding theories of aging. So could we really simulate a brain getting older with a true generative model? So can we simulate aging? This is Thomas Bayes. He was a reverend in the Church of England. Didn't really publish anything until posthumously. It was published by his friends. But he has this famous Bayes rule, which you all know, which says basically, my posterior belief on something theta, pretend that's the weather, given some data, is proportional to what I'm seeing right now, the data given my belief times the prior belief. So if you always think that it's going to be sunny, and you see a few clouds in the sky, well then your belief is that it's probably going to be sunny with a few clouds in the sky. You're less likely to think that it's rainy, but if you have a prior knowledge that it's rainy, you're going to probably assume that it's going to rain any second now if you see a few clouds in the sky. So this is a very it's just a description of conditional probabilities. But it might be fundamental to understanding how the brain works. As you can imagine, as we get life experiences, our priors get certainly more developed as we have more experience with our environment. So what if knowledge is hierarchical, and what if aging builds stronger priors? So how would this evolve or emerge from a brain? So if we know that, remembering this hierarchy, that we get forward connections and backward connections, in the theory of predictive coding, what we're seeing in the backward connections are your prior predictions. So what do I believe basically comes top down. What am I seeing right now in an error message comes bottom up. So there's error propagations that go up the cortical hierarchy, and there's prior predictions that come back down the cortical hierarchy. So my idea of aging is that as you get older, your top-down effects are far more predominant than your bottom-up effects, because you just have more priors, you've been around longer, you're a better Bayesian. This is a statistical framework that's quite detailed, in some respects developed by Carl Friston, who's written about predictive coding in the context of message passing between cortical areas, which can be deciphered with the dynamic causal models, which I've shown you already. And here the idea is the same. So this is just from a Nature Reviews Neuroscience article, which shows you that the forward prediction errors can be stopped by backward predictions. So if I have a well-modeled prior on my environment, all I need to do is send backward connections back, and then that's all my brain has to do. It can predict the world. So what if in a young brain we have lots of forward prediction errors, because we don't have very many priors, and fewer backward predictions, whereas an old brain might have fewer forward prediction errors and much more backward predictions, or better priors. So I'm going to give you some support, at least from neuroimaging, for this sort of scheme in aging brains and changes over the lifespan. So could there be these predictive codes in older brains? So I'm going to show you now, again, this sensory mismatch negativity paradigm, where we show this, we play rather sounds into people's ears that are this beep, beep, boop, beep. And here I'm looking at a lifespan cohort aged 19 to 83. So 98 individuals, they're all healthy with good MMSE scores. And I want to look at the evoked responses and see if they're synonymous with the predictive coding account of aging. And so here in red, I have the brain response to, you've seen this already, the normal tones, and in green is the oppo, you get this MMN effect. Now we know that the MMN is reduced in older adults. So why is it reduced? Is it just there's some discommunication generally, or is it specific to certain cell interactions that might signify these predictive codes being present in older brains? And so the predictive coding interpretation of the mismatch negativity is that neural ensembles deploy top-down predictions of the standard tone. So then when you get an oddball, you get a large prediction error conveyed through forward connections. So you want to change your prior expectations, perhaps if you've seen some in the environment and you need to relearn your environment. However, we are looking at this from an aging point of view. We're interested in where the changes in connectivity were 5 months. And what we saw was our standard regions were activated for all our 98 subjects. And interesting, even though these DCMs have lots of parameters, we just saw one that correlated with the age of our participants. And this change in strength of the forward connection, or the oddball prediction, which was higher for the younger people than it was for the older people. So the younger people had higher predictive errors than the older group. So this, we said that older brains was less susceptible to short-term Bayesian updating, and there was reduced sensory engagement or entrainment. And this, we believe, could be synonymous with reduced model complexity. So as you get older, your brains can generalize better, so it doesn't really distinguish exactly sensory features of the environment, but it more categorically suggests that your aging brain can say, yeah, that's just a tone, it's something played into my ear, rather than overlearning statistical oddballs that might not be behaviorally relevant. However, there's no free lunch as always, so if our Bayesian brains as we get older are better generalizers, so we're able to deal with problems more effectively because we can see what's coming down the road a little, what don't we get? And this is another study that looked at this same sort of bottom-up versus top-down dichotomy in aging, and again found that there was increased activity in top-down effects for older people that was related to worse memory recall in an episodic paired association task. We looked at a dynamic causal model of frontal cortex and found that there was particularly changes in the frontal inhibitory interneuron system in the older brains that might show us why there's increased top-down effects in older brains. Now finally, this is just a recent study we're quite excited about. It's an electrophysiology EEG study on priming. So this is a nice task. You do it in patients as well as older controls. And it's relatively easy. I'm going to get you all to help me on this now. So you're all awake? Okay, can you name each picture as quickly as possible? Okay? This is sort of tricky. I don't know why there's a boulder. Yeah, so crisps, chips. Okay, so if I measured how fast it took you to respond, even if you had Alzheimer's disease, if you're old, if you're young, you will show a repetition priming effect. So you'll be faster, your reaction time will be lower when you see this kitten for the second time. You don't actually have to be able to consciously recall that you saw the kitten earlier. Indeed, in our data, dementia patients cannot recall that they've seen it, but they show a behavioral facilitation. So what is responsible for this naming circuitry? Here I'm showing you younger and older adults, 23 and 21, and I totally agree that older versus younger is poor, it has an impoverished view of aging in many ways, but this was to try and look at the predictive coding models again. So you can see a lot of activity in frontal cortex and occipital lobes and temporal lobes. So, as I said, the behavioral priming, though older people are slower overall at naming the objects, this priming effect is identical to the younger people, so the difference between the kitten and the sunflower, the second kitten and the sunflower. So we both see this behavioral effect, and remembering our predictive coding framework, we want to look at now the interactions between these different brain regions. This is just something to introduce that we were particularly interested in how signals could get to the front of the brain without going through the hierarchy. And we found this interesting literature from Fear, Joe Ledoux, that shows that there's actually inputs that go into the amygdala and orbitofrontal cortex that come directly from the thalamus. So you can sort of bypass all the hierarchical cortical processing. This is also shown in work by Mashi Labar, who's shown that object recognition can go to the frontal cortex first and then go back to the temporal cortex. So it sort of breaks the hierarchy idea of sensory processing. And what we found, we modeled these four regions as a dynamic causal model. And we tested two models, one where we had inputs into the early visual areas only, and one where we had inputs into the early visual area and into the frontal area. And this is our observed data, and this is our model data. So you can see overall these models do pretty well at fitting the event-related potentials that we observe on the scalp. But most interestingly, this is the key finding here of the talk really, is that what we found was that younger people had this selective preference for models where the inputs excited just the early visual regions, whereas in the older population, they preferred a model where you had excitation directly to the frontal cortex. And indeed, the stronger this frontal cortex activity, the better reaction time you had overall. So it's almost like older people are seeing with their prefrontal cortex, or they're predicting what's about to come next and filling in the details later. So predictive codes were also found in the ventral stream from early visual to anterior temporal cortex, where we showed this typical suppression of errors once you see the picture again. So this is the novel item, and when the item is repeated. And in older and younger adults, you saw it along the same pathway, but in slightly different mechanistic regimes. But overall, the take-home is that aging is accompanied by a greater reliance on top-down predictions to suppress bottom-up errors, or that's my casting of the PASA and the Harold arguments, and that the strength of input to frontal cortex in this particular population was found to correlate with behavior on our priming task. And that classical predictive coding effects were found in the ventral visual stream for younger and older adults, but the mechanism was a little different. But overall, I think DCM provides a means to assess network changes over tasks and with age. And we're now also using these paradigms to explore connectivity changes in neurodegenerative disorders, particularly in Alzheimer's disease and in these tasks with EEG and priming, where you get a minimal behavioral and cognitive effects. I'd like to thank my lab and my colleagues and you all for your attention. Thank you again.
Yeah, good combination. Congratulate you. There are questions. Yeah, here, Arun, you were the first one, right?
So, the data that you use to infer that there's predictive coding, how would you distinguish that from, say, for example, differences in attentional engagement between younger and older subjects?
Yeah, great question. So what we do in these tasks is write down a description of predictive coding mathematically and then change the predictive sequence of events and look for changes that are really well modeled by free energy minimization. So the difference with an attention mechanism is sort of almost not there. Maybe that doesn't make a lot of sense. But the idea of attention under this framework is that you change the size of the error that's propagated up the hierarchy. So attention in this setting could do the same thing as a predictive coding scheme. It just has a different name. So it would be the precision of your error. So if you're attending to say this chair is black or something is not moving and then it suddenly moves, your attention is there, but you're also getting bigger errors there. So they're hard to really separate because attention lives in this framework quite neatly. So then it's just a matter of whether the predictive codes sort of show a cancellation as you learn. So that's really the main mechanism by which you can tease them apart. Our neuropsychological assessments were the best we had in terms of early onset or MCI status. They didn't have those indicators on the MMSE. Yeah, they could be. I mean, yes. The only way to test that is to look at potentially a PET score or a tau level, which we haven't got in these cohorts. But it's we need more money to do that.
Yeah, I get the last question. Yeah.
First of all, sorry. First on knowledge.
Yeah.
And in fact, when you look at the semantic processing of words, the top-down comes from the infratemporal because that's where the semantic knowledge is. So in doing those predictions, well, we probably have to be very much task
I agree. Yeah.
oriented.
Yeah. And like you say, if you can see semantic predictions coming on better as you get it older, indeed the top-down activity might be affecting other parts of the brain. You may not see it in an fMRI up in prefrontal cortex, like you say. So I think they are very task-dependent.
Yeah.
Very interesting.
Thank you.
Very good job. That was mathematical microscope. We had heard electronic microscope, and now we have a new term. The third speaker in this session is Dr. Manjari Tripathi, Professor of Neurology. May I invite you into the chair, madam? I'm happy to state that we were alumni in the same institute. We studied together in the same institute. And she has a brilliant track record, working now as the Professor of Neurology at the very prestigious All India Institute of Medical Sciences, New Delhi. And she has had to her credit several publications, several awards, and she is the NIH Fellow at the University of California in Los Angeles. I now invite Manjari to take over and present her paper on risk factors for dementia, role of stress and social indicators. Okay, madam.
Thank you, sir, for the very kind introduction. And we move on from predictive coding to modeling to more earthy topics. And I would like to thank the organizers, Dr. Vijay Lakshmi, for adjusting me here because I was unable to be here yesterday, though I'd had loved to because of the most crowded outpatients which All India Institute sees. it's kind of a paradox when you come to meetings versus the clinical work. I'm just going to be talking about risk factors for dementia and the putative role of stress and social indicators, which has found an increasing interest in the recent past. We know that it's no secret that we are living longer thanks to the cardiologists who are kind of keeping us alive for a long time, and better medical facilities. The aging population in our country is increasing, as you can see here. This is a known fact by the talks yesterday. And India has now the third largest aging population, as can be seen here, following United States and China. And hence, it's for us as clinicians and as well as researchers and neuroscientists to be interested in this as a research entity to prevent the burden faced not only by the patient but the caregiver and the family with this disorder. This is just to show you again the putative rise in the number of cases. And the importance that this has been, it has caught the fancy of even political leaders, is the fact that at the G8 summit in 2013, the priorities for research and investment was thought to be research to elucidate mechanisms underlying the initiation and progression of neurodegeneration, which is what all our modeling and all that is doing. And of course, the second was the prevention of dementia, which you really don't get to until you know the risk factors. And what was seen there was that unless there is a sufficient political will, and of course a research will, as this institute has, where we need to better understand risk factors in predating the disease in younger generations, we will not be able to reduce risk and develop and implement rigorously designed public health programs, which is what we as humans would want to do. And hence, we must take into account tailored national and local needs to prevent these kind of issues which are increasing in our population. But really one, as youth really doesn't know what risk is until, it hits you really hard. And the risk kind of accumulates as you grow along your lifespan. And just for those, because this is a very mixed audience, what is this risk that we are talking about? Now, risk is an attribute that the person or a population has, which is present before the onset of an event, whatever disease it may be. Here we are talking about dementia. So it's an attribute or a characteristic of a person or a population. And of course, you can decide on the risk based more on the incident cases or the new cases rather than the prevalent cases, because the associations with prevalent cases of a particular disease may actually be bringing in an inherent bias to your risk assessment. And if both risk factor and disease are observed at the same time, it again brings a bias, because you really don't know for how long before each has been present. We as clinicians talk of this risk in terms of two terms. One is the relative risk and the other is the odds ratio. And the relative risk is what we calculate in our cohort studies, and if the figure is more than 1, then the risk is harmful, and if it's less than 1, if the risk is, or the factor is protective. The odds ratio is what we talk as epidemiologists and clinicians, which we calculate in case-controlled studies, and generally this is a good thing to calculate where the incidence and prevalence of disease is low. As in our country, we do not have that high a prevalence and incidence, though we really need good epidemiological studies as of now. Wherever you have a low prevalence of a disease of less than 5%, it's good to have an odds ratio on the statistical sample that you have. An odds ratio of 2 would state that there is a twofold increase in your having the risk, that is 100% increase, and that is doubled. And if you see an odds ratio of 1.2, it would just mean a 20% increase, not a 100% increase. So that's how you interpret your odds ratios. Now, how do you decide that this particular factor is definitely a causal risk factor? The only way you really decide that this is a causal risk factor is to have the proof of the pudding, that is to prevent it once you intervene. So randomized controlled studies, large randomized controlled studies, where the risk factor, treatment or prevention of that gets rid of the disease, is the sure short way to say that this risk factor is a causal risk factor. Otherwise, we end up saying there are probable causal risk factors, that is, they are studied, what we studied by the case control studies, like you could say vascular dementia is related to many of these causal risk factors. Until you intervene in these, you would not say it's a definite risk factor. And what's an independent risk factor? Wherever you do multiple logistic regression analysis, and wherever you have an odds of greater than 3 in case control studies and greater than 2 in cohort studies is taken to be an independent factor. And then you have population attributable risk, which is the risk which our population has. And you know India is currently sitting on an epidemic of diabetes and hypertension. And if you talk of numbers, India is probably leading in all the numbers, whether it's the conventional infectious diseases or even the emerging neurodegenerative diseases. And if you talk of a disease like Alzheimer's disease, it's pretty complex. It's got split personalities, it's got multiple facets to it. It involves the realms of activities of daily living, behavior, and cognition. And there are various theories, and people have tried to look at what exactly is causing all this tau and amyloid beta in the brain, whether it's something to do with your genes, whether it's something to do with your biologic mechanisms, the chemistry, or, you know, there are different things which are going on, and there's a recent interest in how we live, the way we express, the way we interact, in which environment, social indicators, stress, etc. have a role to play in this disease. And what exactly converts us from this healthy state of our existence of, you know, being alive and speaking as an audience or listening in an audience, or sleeping or whatever, the diet we eat, and ultimately resulting in dementia is still a mystery and enigma to many of us, and most of us in the clinic really look at it in form of unmodifiable risk factors, some of which are really genetic, and they're not too many, but genetic risk factors have been discussed yesterday by Dr. Sudha, so I'm not going to go into that. And of course the age, increasing age. Now these two are pretty unmodifiable. They're something we really can't do much about until really something comes up in the genetic therapy aspect of whatever we see. And of course, the clues to this genetic onset is the early onset of dementia, and of course, a strong family history which many of our patients may have. There's something known as a lifetime risk also, and if you're a woman, you have 1 in 6 women have the chance of having the condition, and 1 in 10 men. So you know you have inherent lifetime risk which is increased in women. And of course, these are the 7 sins which, if you have any of these, diabetes, midlife hypertension, obesity, physical inactivity, depression, smoking, and low educational attainment. And of course, various environmental factors which have been enlisted by Martin Prince Group and Norton's Group. Exhaustively, we know that if you have these 7 sins within your life, you are more likely to be having dementia, etc. And then Sudha's work on elevated plasma homocysteine levels, low serum B12, folate, all these are probably various factors which we find in many of our patients. Now, all these risk factors have been looked at in meta-analysis, which is probably the purest way of looking at various data points which we have collected in various studies through our human lifetime. And then these lifestyle factors which I mentioned, obesity increases the relative risk 1.4. Smoking, of course, again 2.2. Physical activity, the more you sit, sitting is the new smoking. In fact, many meetings should be held standing, because what risk smoking poses to you is probably 8 hours or more of sitting, and probably labs where there are no chairs, a better environment. So lack of physical activity, if you have physical activity, you're protected. If you have a good cognitive reserve, whether it's intelligence, occupation, and education, which all of you must be having here at the Indian Institute of Science, you have a good cognitive reserve and can prevent dementia. And of course, bilingualism, multilingualism, which most of us are here in India. Alcohol intake offers a moderate benefit, provided you know when to stop it. And then, of course, medical conditions, midlife hypertension. Now look at the number of studies. There are huge studies with huge populations involved. Stroke, again, large studies which say there's risk, diabetes, midlife, increased cholesterol, again. And then very few intervention studies. So I said the proof of the pudding lies in intervention studies. But you look at the intervention studies, the only study which has been positive is intervention on hypertension. So if you intervene on hypertension early, you're going to reduce your odds of having dementia. But intervention studies with statins and B12 and folate really haven't yielded the same results which we would expect. In India, we know that the prevalence rates are not so great as one would like to see in the West. We're talking much more about it, and you can see here it's about 3 to 3.5, and there are some studies. Our own center has undertaken a longitudinal study, which was started in October this month for 10 years, and Sudha's on board, where we are doing a longitudinal follow-up of 2 cohorts of people. One is from an urban area in Delhi, which is Vasant Kunj, and the next is from Manesar. And these people are going to be followed up for 10 years. They are normal, healthy, 50-plus adults followed up for 10 years to see where they develop cognitive. So the purpose of our study is looking at cognition and stroke, and this study is headed by the chairman of my department, Dr. Prasad, and we will be, going along the study and yielding results. But before that, we looked at our population, which presents to us at AIIMS, considering the fact that we have not too many patients out there in the field for dementia. We thought we'd do a risk factor analysis, and we'd try to find out if there's anything which could protect our patients or put our patients at risk. And so we looked at multiple factors in our population at AIIMS. And this was a study which we conducted over 300 patients and looked at various risk factors which were prevalent. And you can see here the multivariate logistic regression of our North Indian population clearly showed that participation in cognitively stimulating activities, which we had defined in terms of hours and number, was protective. And of course, eating vegetable oils which were rich in PUFA, which not many of our population does. Most of our population still lives on dalda and highly saturated fats, was again protective, and eating salads was protective too. But then urban living increased the odds. Remember, I said any odd above 3. So urban living itself increased the odds to above 3. Lack of exercise increased the odds to about 2.8. And of course, diabetes, depression, and saturated fats increased the odds to quite some high. And what we found interesting was pickles, because in India we consume a lot of pickles. And what are these pickles? These pickles are nothing but fat and salt, which are going to directly or indirectly increase your hypertension, was also increased with an increased odds of 3.9 for dementia. And of course, the ApoE phenotype, which we had previously published, an increased BMI and increased cholesterol had an increased odds. So what we found from our population in North India was that there were certain protective factors. If you're going to be socialized and interact with your peers and seniors, get your blood pressure checked in a pretty aggressive manner. Some people do it daily, mind you, and eat good green stuff without getting neurocystic in our country, and monitor your sugar and have a good physical activity, as well as do a lot of networking and learn new things. Even learning computers at old age is going to prevent dementia in our population. And all the usual, processed foods, obesity, butter, etc., smoking, depression are going to put you at risk. So with this in mind, we then thought we'd look at the number of patients who come to us, just the early Alzheimer's, that is the CDR of 1, and that's very difficult to get by in our clinic. Many of them come in stage 2 and 3 to us, or they come in MCI. So we looked at stress, contribution of stress as well as in our population, and there are varied definitions of stress, but if you have any of these things, agitation, poor judgment, moodiness, general negative outlook, irritability, excessive worrying, be sure these are signs of stress and recognize them early, because they could be a harbinger of increasing your risk for dementia. And basically the stress is caused by multiple demands or stressors, such as an inadequate fit between what we need and what we are capable of. So this mismatch of what we of our existence is probably responsible for a lot of stress. Now, stress works by various mechanisms, and the neurobiology of stress itself, or psychoneuroimmunology itself is a fascinating field, but it does affect our hypothalamic neurons, affects the CRH, the ACTH, cortisol, and of course there's this stress which builds up. How does it affect the brain? And there are various studies published of late, even in biological psychiatry, working both with experimental models of rats as well as human populations, which have clearly shown that stress probably activates this HPA axis and increases the glucocorticoid hormones, leads to neural degeneration, cognitive decline, and dementia. So this is the mechanism which has been looked for in all these studies done by various authors. And hence we applied 2 stress scales which are standardized for the Indian population, which is the presumptive stressful life event scale and the depression anxiety stress scale to our population, and looked at various parameters using multiple logistic regression method of analysis. And what we found, and our definition of dementia was of course based on the DSM-5 as well as the NINDS-DRDA criteria, and we just took a CDR of 1 because it's not easy to assess people who are moderate to severely affected. And we have a population of 55 so far. The study is ongoing, and of course we found higher stress scores in patients as well as caregivers, and this was significant. We also found higher depression scores in patients as well as their caregivers as compared to the healthy aging controls. Cortisol analysis, again, we find a higher value of the same in the active, that is the CDR1 dementia group as compared to the reference group. And ACTH wasn't much different, but again, it was different when compared to the controls or the normal aging population. Prolactin, again, was different when we compared the dementia CDR1 to the related references, but not with the non-related references. And then we also look at sleep in another of our patients who had, this was a different set of patients. So we have one group which is being looked at for sleep and the other for stress. And we know that sleep has an important role in consolidation of memory, especially the non-REM and REM both play a very important role. And apart from doing the objective sleep assessment by the polysomnography, we are looking at Pittsburgh Sleep Quality Index and Epworth Sleep Scale, and of course a huge difference in between patients, mild Alzheimer's to the caregivers, which is significant, and the Epworth Sleep Scale, again, there's a huge difference saying that sleep there are sleep issues in our early patients, and they may have even predated them. Personality, and we know that personality predicts longevity, especially how conscientious you are, how physically active you are, and if you are in more feminine occupations. And of course, those who are conscientious have better health behavior, less risky behavior, happier marriages, better friendships, and optimum work setting. And this leads to decreased catastrophizing and life satisfaction. There are several studies which have come off late in 2014, sample size of 500. They've looked at these personality issues and found higher degree of neuroticism in patients who have Alzheimer's disease. Again here 2013, higher degree of neuroticism in patients. And 2009, where again a different scale was used, and it was again found to be the same. Now what is this neuroticism? And many of us may know people who have a characteristic personality of anxiety, fear, moodiness, worry, envy, frustration, jealousy, loneliness. Now these are very descriptive terms, and if the scale helps you objectivize it, and if you can apply it to your population, it's an interesting way of looking at population behaviors and how you can actually build reserve and plasticity of the brain based on converting from neuroticism to extroversion. And extroversion personality has a less trend towards dementia. In our patients, we had about 42 we have analyzed so far. The study is ongoing. You see the age group, we have around 60, the active is around 64, and most of them are men for some reason. We get more of men coming with this problem, and most of our patients are married, very few divorced, 1.6%. Most of them belong to the middle class, so it's not the upper or the low, it's mostly the middle class which is coming. And then we have looked at these social indicators in this population and looked at the neuroticism scale, looked at extroversion, life satisfaction, spirituality, and loneliness, assessed by the UCLS loneliness scale. And definitely there is a trend towards higher neuroticism in our MCI and Alzheimer's. So this included MCI patients also, a lower extroversion. Again, this is higher in the patient group, and of course loneliness. And cognitive health probably begins much more earlier in life, and it is an ongoing process rather than something which develops at the age of 60 or 65, and probably intervening right from the time you're born to the time you're probably incapacitated, whether it be effects of how you're brought up as a child, how socially engaged, you're in an environment, what kind of diet and nutrition you follow, what kind of exercise routine you follow, how long you sit on a chair, whether you're depressed, and manage all those kind of risk factors which develop in midlife, go a long way in reducing the risk of cognitive decline in our community. And with that, I'd like to end my talk, and if there are any queries, I can answer.
Remain healthy and free from dementia. You supported my observation that yoga was beneficial to the brain. Okay, it's open for questions. Yes, madam. Yeah, here. There
So you, so you showed, how the levels by questionnaire. I think there was reportable questionnaires were higher in mildly impaired or demented patients. How do you tease apart from the very fact of the illness increasing their stress levels?
Yeah, so this is an inherent bias one taking in patients, and you're not doing a longitudinal study, or you're following healthy elderly people. This kind of bias is going to creep in because how far the patient is going to report and the caregiver is going to report on that particular scale is a problem. However, as I said, the longitudinal study we are undertaking, that will take into account a prospective follow-up of the healthy aging population. So you will get a more definitive answer from there. But the fact remains that when you compare it with the caregivers who are exposed to the similar environment, which of course the patient is in, it is significantly more. So that's all you can understand from that. But then, as you say, there is an inherent bias in this way of analysis.
An attempt at levity, though. It seems marriage is an indicator for stress.
There are several studies which have looked at marriage and cognitive decline, and it is postulated that your longevity is more if you're married, and you're less to have, you're more likely to have a better cognitive reserve if you having a companion rather than being lonely. So I think that is more important than just the concept of having some companion as you age.
I have one question. Depression itself is amnesia and decreased cognitive performance is a known problem in depression. So is this?
We talked about mathematical microscope, how mathematicians can help us identify the regions of brain where it's pathological in degenerative disorders. And of course, we had a very stimulating talk about how we can continue to keep using the brain and not lose it. I congratulate all the speakers. I thank all of you and close this session. Thank you.